lymphocyte development T cell will B cell. Objectives: compare and contrast the origin and maturation of B and T cells. بعدين understand the role of primary lymphoid organs in lymphocyte development. Compare the mechanism of positive and negative selection negative in lymphocyte development. بس لحظة. تمام. Understand the gene rearrangement of TCR and BCR. Recall, what is the difference between the primary lymphoid organ and the secondary lymphoid organ? Where do B and T cell mature? هسه رح نعرف. Primary lymphoid organs are two, bone marrow والthymus. They are the source of naive lymphocytes, B cells and T cells. There are the sites which lymphocytes form and mature. Both B and T lymphocytes arise from the same source, which is the hematopoietic stem cell. وين موجودة هاي hematopoietic stem cell؟ موجودة بالبون مارو. تمام. B lymphocytes remain within the bone marrow during the process of maturation. مو بس بالبون مارو، هم يعني secondary lymphoid organ تتدخل. T lymphocyte migrate to the thymus to mature and differentiate. The secondary lymphoid organ includes the spleen, the lymph nodes, المالت, which is the mucosa associated lymphatic tissue, الجالت اللي هي الgut associated lymphatic tissue. It includes Peyer's patches, nalt in the nasal cavity, balt in the bronchial. In the secondary, this is where the antigen presentation occurs. Antigen travels to the secondary lymphoid organ carried by an APC antigen presenting cell. In the secondary lymphoid organ, where it's presented to the lymphocytes, the site of differentiation of mature naive lymphocytes into effector cells. This is where the cells, the lymphocytes, proliferate and differentiate into either effector cells or memory cells. نجي هسه على pattern of B cells and T cells. هسه B cells, they are formed in the bone marrow. They start the maturation in the bone marrow, بس إنه it doesn't complete the maturation there. It leaves as immature, travels to a secondary lymphatic organ, which is the spleen, and complete the maturation there. هاي بالنسبة للB cells. T cells, الفورميشن مالتها production starts in the bone marrow, وبعدين it travels to the thymus, where it starts and complete the maturation process. هاي الفرق بيناتهم. نجي هسه على thymic education. Thymic education are important for two things. Number one, generate a diverse pool of specific T cells, وهاي العملية تستمر من الولادة لحد البيوبرتي. This is in order to achieve the immunocompetence. Immunocompetence, where the lymphocyte, the T cell, have the T cell receptor that is specific for a specific antigen. Second purpose, ensure that self-reacting T cells are not produced, اللي هي self-tolerance. فهسه thymic education are important for two things. Immunocompetence, it includes the diversity of specific lymphocytes to specific antigens, و self-tolerance بحيث انه ما يصير اكو autoimmunity. نجي هسه على thymus. This is the thymus. It is most active when we are born. Yes, it is more active when we are born, and when we reach 10 years old starts to atrophy. نجي هسه histology. الثايمس مثل ما موضحة هنا contains of two parts, the cortex and the medulla. الcortex, it is a dark, يعني لما نحط dye, it appears as dark. Why does it appear as dark? Because it contains densely packed immature T lymphocytes, or the thymocytes. In medulla, it is lighter. Because it contains fewer cells, and these cells are mature lymphocytes. What are the cells that are present in the thymus? عندك thymic epithelial cells, dendritic cells, والmacrophages. هنا الثايمس الفانكشن مالتها, it is the site of maturation and differentiation of lymphocyte. مو البرودكشن in the bone marrow. Maturation and differentiation happens in the thymus of the lymphocytes, of the T lymphocytes. Thymus undergoes age-related thymic involution. إنه it atrophies. In neonate, thymus is the largest and most active. In adults, it's atrophic and it has been replaced by adipose tissue or fatty tissue. زين هسه احنا راح نتكلم على المaturation مال T cells. First of all, during the 8th week of gestation, hematopoietic stem cell, which يعني the source of hematopoietic stem cell وين موجودة بالbone marrow, it migrates to the thymus. The maturation of the T cells involves the TCR rearrangement in order to form the TCR receptor and exposure of surface molecules اللي هي CD4 positive, CD8 positive. Thymus creates an environment for T cell maturation education using positive and negative selection. هسه راح نشرحها. وبالأخير what is produced from the thymus are mature naive T cells that are immunocompetent and self-tolerant and will migrate to peripheral lymphoid organs like the lymph nodes, tonsils, MALT, GALT, NALT, BALT in order to encounter the antigen. و T cell repertoire اللي هي الستوك أو الريزرفوير it's completed at puberty. شنو المقصود بهذا؟ المقصود إنه قلنا احنا الثايمس is most active in the newborns. فهنا maturation differentiation of the T cell happens يعني at the highest rate. The pool of different and diverse T cell that are specific to antigen are formed till puberty is complete, till puberty. وبعدين what happens to the thymus? It decreases in size, its function decreases, وبالأخير يعني توقف. عندنا هنا دي يوضح لنا ال T cell receptor. T cell receptor عندنا MHC class 1, MHC class 2. MHC class 1 binds to which one? Binds to CD8 cells اللي هي راح تصير بالأخير cytotoxic T cell. لاحظي هنا ال MHC binds to the TCR, T cell receptor, and the CD8 is a co-receptor. It enhances the binding, facilitate the binding between the TCR and the MHC, and does signal transduction in order to activate. And the MHC2 class 2 binds to CD4 cells. وهنا دي واضح لنا انه CD4 CD8 increase the sensitivity of T cells to peptide antigen complex by 100-fold. فلهذا it facilitates the binding between these two مثل ما قلت. هسه نجي على ال development of the T cells. طبعا احنا قلنا انه thymus consists of an inner وين لحظة ما ادري صار. المهم قلنا انه الثايمس it presents يعني it contains two parts, the cortex والmedulla. The hematopoietic stem cell enters the cortex and through the يعني journey to the medulla it matures. هسه hematopoietic, as you can see here, enters the cortex. It becomes what? It becomes the lymphoid progenitor. Lymphoid progenitor, then it develops into DN1, DN2, DN3, DN4. This DN stands for double negative. Double negative, it does not contain CD4 or CD8. And during this double negative stage, TCR rearrangement occurs. تمام؟ هسه احنا شغلة لازم نعرف عن T cell receptor انه it contains two chains, alpha chain and beta chain. Each chain consists of two sites, two regions, constant region or variable region. The TCR rearrangement, which happens at the DNA level, it basically codes for the variable region of the chain. This variable region of the chain, which makes it specific to a specific antigen. يعني specific affinity to a specific antigen. فهاي التي سي ار rearrangement. زين شو يصير بالتي سي ار rearrangement؟ هنا عندنا بالدبل نيجاتيف T cell يعني during development we have a germline DNA. It consists of three regions: V region, D region, J region. V stands for variable, D stands for diversity, and J stands for joining. اللي يصير there are enzymes that cut specific regions, cuts a region from this, cuts a region from D, cuts a region from J. بالاخير what happens these يعني هاي الاجزاء اللي تمت يعني اقتطاعها it will join again, it will undergo somatic recombination in order to form new يعني new VDJ region specific. احنا مثلا احنا عندنا هنا V1, احنا عندنا V2, احنا عندنا هنا VN, ممكن يكون V5 V6. الامر اشبه بين انه يعني choosing an outfit. You have for example a cap, a t-shirt, a trouser. You have like 5 trousers, 5 caps, 5 t-shirts. It is a random process that you choose the cap, the t-shirt and the trouser. يعني يصير random. There are lots of possibilities, تقريبا يمكن 125 possibilities. اما هنا من ناحية الدي ان اي عندنا ملايين, millions of possibilities. فبالاخير what will be generated is a specific VDJ combination, somatic recombination, يا اما هذا يا اما هذا يا اما هذا. And this VDJ will code for what? For the variable region of the TCR, for the TCR receptor مثل ما وضح لنا. زين. And this process TCR rearrangement, where does it happen? It happens at the double negative stage. هاي DN1, DN2, DN3, DN4. ورا DN4 اللي يصير انه الجينز starts coding for CD4, both CD4 and CD8. فبالاخير راح يكون عندنا double positive DP cell double positive. This double positive consists of what? خلينا نرسمها هنا, حاول ارسمها. خلينا نفترض انه هاي double positive. It contains what? It contains the TCR receptor. This is the TCR receptor, CD4 لا ما ادري شلون, CD4 و CD8. خلينا نفترض انه CD8 T shape, CD4 هاي على سبيل المثال. This is the double positive. هسه اللي راح يصير انه اكو عندنا cortical epithelial cells. Cortical epithelial cells have both class MHC1, class MHC2. فهنا راح تتقرب cortical epithelial cells which have both classes of MHC. راح نفترض ان هذا MHC1 وهذا MHC2. ليش صار MHC2؟ هسه اللي راح يصير انه راح تتقرب الى هاي الدبل بوزيتيف سيل. if there are inadequate number of TCR or co-receptor اللي هي CD4 CD8 signaling inadequate يعني عدد قليل, هاي ما منها فائدة T cell, so it will undergo apoptosis. اما اذا كان اكو هناك adequate TCR و adequate co-receptor signaling, راح ننتقل للخطوة الثانية اللي هي depending on the double positive cell, depending on the higher affinity, يعني it will choose into binding to either MHC2 or MHC1. It's a random process. ممكن يعني تعتمد على affinity. If the affinity between the CD4 and class 2 is higher than the affinity between CD8 and class 1, هنا it will bind to class 2 MHC وراح تصير عندنا CD4 committed double positive. هسه احنا عندنا انه CD4 has binded to the class 2 MHC. شي يصير CD8? It will be downgraded down regulated أو بالأحرى الجين that codes for CD8 will be suppressed وبالأخير CD8 will disappear from as a cell surface marker اللي راح يبقى CD4. فلهذا we have CD4 committed double positive. It contains both CD4 and CD8. Why? We have double positive. بس بالأخير down regulation راح تكون لنا CD4 positive single positive SP. هنا it doesn't contain CD8. ونفس الكلام if the double positive cell has a higher affinity to class 1 MHC protein, it will be CD8 committed double positive. CD4 will be down regulated وبالأخير راح يتكون لنا CD8 single positive. زين احنا هسه خلصنا أول purpose of the thymic education which is immunocompetence. عندنا ال TCR receptor that is specific. باقينا شنو self tolerance. This is for the positive and negative selection. هاي كلها اللي تكلمت عنها انه بالخطوات والى اخره. هنا كاتب the type of CD that the T cell has higher affinity for is kept while the other is down regulated مثل ما قلت. هنا بالنسبة للpositive and negative selection ال TCR هسه الانتيجين self antigen that is presented on MHC protein T cell receptor binds to two regions, binds to the antigen, to the self antigen and binds to the groove. هسه عندنا احنا هنا self antigen هذا self antigen وعندنا الجروف. The TCR binds to both of them. The proper binding, which is basically the positive selection, it doesn't recognize the self antigen. This is number one, and binds properly to the groove of MHC. This is for proper binding or positive selection. زين نجي هسه على positive selection, negative selection. Negative selection, it will die. The fate is apoptosis. The positive selection, it will be kept. Now, if the T cell recognizes the antigen, no matter of the binding of the groove وإلى آخره, the outcome is death. زين. If the TCR doesn't recognize the self antigen, هاي حققنا أول شرط. الشرط الثاني البايندينج very weak binding to the groove احنا ما نريد ما راح يكون منها فايدة ف it will die. ونفس الكلام if the binding was strong. If the binding was strong, it will also die. The positive selection, first of all, it doesn't recognize the antigen and binds to the groove with proper affinity. If it binds to the groove with proper affinity, the outcome will be CD4 or CD8 اللي هي هذه. CD4 positive single positive, CD8 single positive. أما إذا it doesn't recognize the antigen but has moderate affinity, low recognition, it will form T regulatory cells. هاي البارادوكسال سelection هسه اللي تكلمت عنه. نادي وضح لنا انه that in the transition of a committed double positive T cell into single positive, the cell downregulates the unwanted CD and removes it from its surface. هنا example CD8 committed double positive removes CD4. CD4 committed double positive removes CD8 وإلى آخره. هنا دي يتكلمون على in the medulla of the thymus, there is something called the autoimmune regulator protein. It is a transcription factor present in the medulla of the thymus. It regulates the expression of autoantigens in the thymus medulla. الوظيفة مالتها إنه it enhance and facilitate the elimination of self-reactive T cells. فإذا عندنا مشكلة بالautoimmune regulator protein, there will be an autoimmune disease. The ability of getting rid of the self-reactive T cells will be reduced وإلى آخره. وهنا عندنا ال دي جورج syndrome اللي أخذناها إنه defect in chromosome 22. It will result in missing or small thymus. زين هسه missing or small thymus هنا the production of naive T cell will be affected. We will have T cell deficiency. هل هذا راح يأثر على البي سيل؟ نعم راح يأثر على البي سيل. ليش؟ لأنه احنا بالمحاضرة السابقة قلنا إنه most of the activation of the B cell depends on the helper T cell. فإحنا إذا عندنا deficiency in the T cell, how will we activate the B cell? So B cells are produced, but they are not activated. They do not produce antibodies.